Good morning, students. Welcome to this new chapter, Genetic Engineering. Biotechnology Principles and Process is the name of the chapter, chapter number 11, where you will study about the genetic engineering. You must have heard several times people talking about genetic engineering. What is it? It is the latest technology that is applied for many different purposes. So, first of all, we will discuss what is biotechnology. The word biotechnology itself gives you an idea that some sort of technology is used in our biological life. So, this is what biotechnology. It can be applied for human beings, for animals, for the field products, that is agricultural products, and many different areas. So, what is biotechnology? It is the technology that is used for increasing the capacity, capability, nutritive value of all the things that we consume or we use, or we can produce many things which we need in large quantity, but they are not available for us by the nature. So, it deals with the technology using living organisms, enzymes, etc. As I already explained, that the living organisms which are present and we apply some technology to it, then this type of technology is known as biotechnology. There are two phases of biotechnology. Earlier also, since our ancient times, our grandmothers used to use this technology that is known as old or traditional technology. But they did not know that it was actually a technology. They practiced it because they knew about it. And that is where we again comes the curd. First of all, curd, where biotechnology was used since several thousand years. Like we add some lactic acid bacillus to the milk to convert it into curd. So this is the old tradition. Earlier also, people used to prepare wine, vinegar, beverages through this type of technology, that is by fermenting it or adding something to it. So this was also a kind of technology that was earlier used. So, but people did not call it technology because they never knew the word technology. So, curd, vinegar, wine are the old traditional way of using the technology in our food materials to increase its nutritive value, the vitamins, the minerals, etc. So, have you understood what is technology, biotechnology? It is using something extra to achieve something extra. That is, it deals with the technology using living organisms, like we use lactic acid bacillus to the milk. It is a living thing which is used into another living thing like milk, and then we get a product called curd. Now, in modern biotechnology, what happens? We use genetically modified organisms. Genetically, we have modified many of the organisms and now we use it. The most common example is the test tube baby, which you must have heard several times, that this child is a test tube baby. Do you really think a baby can be born in a test tube? No. The thing is that we use some kind of technology, and this is done by the technicians who prepare the first few cells of the baby in a laboratory, in a petri dish, and this cells or these cells, they are introduced into the mother's womb, and where the baby grows. So this is known as in vitro fertilization. In vitro fertilization means preparing the cells, few initial cells in the laboratory with the help of the chemicals, and then using it into for the mother who cannot get their babies in a natural way. So here we are using some technology. That's why this is known as in vitro fertilization, where some technology is being used. This is called test tube baby. Now we come here. DNA vaccine. You must be hearing about vaccines these days, and you have also used these vaccines for yourself in your early childhood. What is this vaccine? Vaccine is a kind of medicine which is introduced into your body. This acts as an antibody for several types of diseases which can be totally eradicated, and then we are safe and healthy. So vaccine is a kind of technology. So here we introduce some antibodies into the body to fight with the disease causing germs, viruses, bacteria, and we are safe. So vaccine, now they are finding out vaccine for the coronavirus, and everybody is busy. All the doctors are busy finding out a vaccine as an antibody for cure of corona. Now, correcting the gene. What is this? If we have you must have seen people getting some diseases from their forefathers. Maybe in a family, every child is a blind child. How this happens? Since ages, their family is getting only blind child. Why? Because somewhere the genes must have gone wrong, and therefore they get a child who is always blind from generation after generation. So we can find out which gene is responsible for this type of defect. Or you must have seen people getting 5 fingers, every generation 6 fingers, every generation they must be having a 6-finger child. So how this happens? This happens because there is some defect in the gene. Now scientists can find out the defective gene from where you can get the gene, you know the position of the gene, and you can remove that gene and place a correct gene, and you will get from next generation onwards, you will not have a blind child in your family, because the scientists have applied some technology and they have corrected the defective gene, and now you are getting a baby who is not blind. So this type of things, these things are called biotechnology, where technology is applied to correct the defective gene. Now, genetic engineering. What is genetic engineering? Genetic means something related to the gene. So, in the genes where the defects are found, they replace the defective gene with a better and good gene or the correct gene. This is known as genetic engineering, which in future, when you opt for the subject genetic engineering, you will study in detail about all these, and perhaps one day you can become a very famous genetic engineer. So yes, the genes, the defective material are altered or changed into perfect materials, and they are applied to the defective place, and you can become correct. Or if you are having some hereditary diseases, you can find out the defective gene and change the gene. The doctors, the genetic engineers will change the defective gene, and you will get a correct gene, and there will be no more of this type of problems. Now, what is cloning? Cloning is also a part of genetic engineering. You must have heard the word cloning several times, or you have read the story about the clone sheep, that is Dolly, or the clone cow, that was Rosie. These animals were cloned from their mother's cell. That means the genetic engineers took out a cell from the mother and they prepared it in the laboratory, and then they cloned. Cloning means making several copies of the same thing, like you do, which in common word you say photocopy or Xerox copy. So cloning is a type of copy. You can copy exactly the same thing that is from your mother, or another animal can be prepared. This is known as cloning. Then gene transfer. What is gene transfer? If there is no gene, some gene is lacking in your body, some type of, then what you can do, you can isolate that exact gene, that this, and you can transfer it, you can take it out, that from your amino acid, the sixth amino acid is not working properly, so you remove the sixth amino acid and you place a good gene over there, and then you can correct the thing. So, isolation and introduction of new set of genes. You can do it from when you study genetic engineering. This will be very easy for you to understand. Now you just understand that a defective gene can be transferred or isolated or removed from your body, and a correct gene can be introduced so that the defect that is in your body or anybody's body can be replaced and it can be corrected. There are many inborn error of metabolism. You must have heard that people having some diseases from getting some diseases from their ancestors. And this can be corrected through isolation and introduction of new set of genes. Now, recombinant DNA molecule. What is it? First recombinant DNA molecule constructed by Stanley Cohen, Herbert Boyer in 1972. So, it was the first recombinant DNA which was found out by these two scientists, Stanley Cohen and Herbert Boyer, in 1972. Then, what type of tools are used for these type of gene recombinant, DNA recombinant? They are enzymes, polymerase enzymes, ligase, vectors, and other organisms. So, when you when you will further study in detail about this biotechnology in your life, you will come to know more about it. Now, in this class, you have a limited, just one or 2 points are there in your syllabus. So, this is a very small chapter. You have understood what is biotechnology. That is more important to understand. And you got a idea of what is gene transfer, isolation, correcting, or about the test tube baby. So I think you have enjoyed this part of the video, and that is the end. Thank you, children, for listening so patiently and enjoying the class.